from Purdue University in 2013. And then he did a postdoc in Professor Richard Caprioli lab at Vanderbilt University, where he actually studied matrix-assisted laser desorption ionization, which is MALDI, imaging mass spectrometry. And Boone has really been a rising star in imaging mass spectrometry. So in 2018, we were able to recruit him to join our faculty in Department of Chemistry. And his research is focused on developing next generation bioanalytical mass spectrometry to better understand the molecular basis of health and disease. And Boone has been doing really, really well. I saw he just got a 5-year R1. Congratulations, Boone. And today he's going to show us how he actually used MALDI imaging mass spectrometry to do metabolomics, and he will also show us some real-life applications. Let's welcome Dr. Boone Prentice.
Thank you, Siju. It's my pleasure to speak with you all today and introduce the concept and workflow of imaging mass spectrometry or mass spectrometry imaging. You'll hear it kind of termed either way, IMS or MSI. And so what I'm going to try to do today is strike a balance in, over the next hour or so, talking about some of the fundamental aspects of imaging, how we do the imaging experiment and how it works at a fundamental level. I'll also try and highlight some of the important experimental parameters that we need to consider when setting up or designing an imaging experiment. I'll also talk about some of the technical aspects of doing the imaging experiment. So I'll try and show a couple of videos and demonstrations of how we prepare samples and run the instrument. I'll also try and highlight some new directions that I think the field is moving in, some kind of exciting avenues of research that our lab and others are pursuing. And then also, as Dr. Chen said, talk about a few applications. I know this is obviously a metabolomics workshop, but in general, this imaging workflow can be applied to a wide variety of analytes and systems. And so I'll highlight kind of here and there some of those aspects. So I like to start by putting this work in context of the field of biological imaging. So we've been talking a lot over the past day and a half or so about kind of hardcore, metabolomics, but I think thinking in an imaging context is a little bit of a shift in gears. And so if we consider the general field of biological imaging, this isn't a new field. It probably has its origins around, the mid to late 1600s when Van Leeuwenhoek first developed the light microscope. And if we consider the progress in the field over the last 350 years or so, there certainly has been tremendous advancement in the technology. So on the left is a picture of Van Leeuwenhoek's microscope. And on the right is a picture of Eric Bedsing's Palm device, for which he won the Nobel Prize in 2014 for super resolution microscopy. And Eric Bedsing's is a fascinating story. If you haven't heard it, he actually built this device in his living room when he was technically unemployed. So it's a really excellent story in perseverance and innovation and creativity. I'm an instrumentalist, so I think that this is kind of fascinating, this technology development, but perhaps more important than that is the improvement in the type of information we're able to glean using this technology. So on the bottom left here is a sketch of some of the microbes that Van Leeuwenhoek observed under his light microscope when he was first performing these experiments. And on the right is a picture of neuronal activity in a zebrafish brain. And I think you can appreciate that the level of detail, structural detail, information content we get in this bottom right image is certainly tremendously more than the bottom left. And that's really important because one of the easiest ways we have for understanding complex systems and for translating or conveying that understanding and information to other individuals is through the use of images. And if we consider the general suite of imaging modalities available to us as scientists, these run the gamut from things like ultrasound, X-ray, MRI, things that you would encounter in your everyday clinician's office, when you go to the doctor's appointment for a normal checkup or a procedure, to things that, maybe in a clinical setting, but are maybe a little bit more behind the scenes, things like stain microscopy that a pathologist might use or PET. to things that are, I would say even more kind of on the research side at this stage, things like tag microscopy and imaging mass spectrometry, which are certainly making their ways more and more into the clinic, but are a little bit still more behind the scenes. And each of these certainly has their benefits, their pros and cons, things that they do very well and things that may be challenging. For example, a number of these are capable of what I would call in vivo measurements. So measurements while the subject is still alive. Others require tags. So these could be antibody tags or radio labels. And this could be kind of an extra step and maybe a detriment to the workflow, depending on what you're trying to achieve. And still others are capable of what I would call very high spatial resolution. So, imaging single cells or even subcellular levels of structural detail. But what I think imaging mass spectrometry can bring to this field is a very high degree of molecular specificity. So we're taking the specificity of the mass spectrometer, and you've learned about that over the past day and a half or so, the immense way that we can use the mass spectrometer to identify all these different types of metabolites. And we're using that instrument now in a fashion that gives us spatially informative measurements. So we're combining this spatial aspect of classical microscopy with mass spectrometry. And I'll talk about MALDI here today, and I'll introduce the concept of MALDI, what it is and how it works in just a minute. But just know that there are a few other flavors of imaging mass spectrometry that use other ionization techniques as well, things like DESI, desorption electrospray ionization, and SIMS, secondary ion mass spectrometry. So why is this important? Why do we need this level of molecular specificity? Well, as this group can certainly appreciate, this can have a profound effect on whatever application it is that you're studying. So the Shah group recently published an example where they were looking at fatty acid 18-1 and the distribution of this fatty acid in normal and cancerous mouse breast tissue. And essentially they were looking at two isomers of fatty acid 18-1, the delta 11 isomer and the delta 9 isomer. And so for those of you who aren't familiar with this type of nomenclature, 18-1 is the total number, the 18 is the total number of carbons in the fatty acid tail, and 1 is the number of unsaturations, so a single double bond present in this tail. And the delta 11 and delta 9 refer to the position along the backbone here. And so you can see that just by changing the position along the backbone of the double bond, this had a profound difference in the relative presence of these isomers in normal versus cancerous tissue. And so this is obviously a very subtle chemical change. These are isomers, so they're the exact same molecular weight, but they differ just by the single position. And so we know then that metabolomics can provide really important information on disease systems. So it can provide biomarkers for identification and classification. This information can also help us understand disease progression and how the disease is kind of advancing or how it may be infectious. And if we can understand that better, we can hopefully develop new therapeutic strategies to help combat disease and infection. So as we've talked about, over the course of the last day and a half or so, most of the metabolomic studies that are used in labs around the world utilize tissue cell extractions or homogenates or biological fluids such as serum and urine. And so this involves taking the analytical sample, extracting the metabolites or lipids or whatever you're going to analyze in some form, typically doing some type of chromatographic separation and then analyzing by mass spectrometry. And so this is kind of what we've just been learning about. But as we know in some samples, spatial context is very useful and can be very important. And so in the field of pathology, this is very, very well known. And so a pathologist may look at a stained tissue section. This could be a biopsy, for example, from an organ or a tissue that is suspected to be tumorous or cancerous. And the pathologist will look at this under the microscope, and they can clearly see different regions within this single small piece of tissue, right? So they might see tumor cell nests, they might see tumor with regions of stroma inflammation, and then of course they may see normal and normal lung tissue. And so obviously if we were to take this entire tissue sample and homogenize it up and analyze it via LCMS or GCMS or some type of classical metabolomics workflow, we would have lost this spatial context when in fact this tissue both has normal and uninfected tissue as well as disease tissue. And so that can be really important if we're, for example, doing a tumor resection surgery or some other type of analysis. And so classical histopathology uses a number of different types of stains to stain those tissue sections and allow for visualization of the cells for the pathologist to make a diagnosis or a classification. And one of the most common stains is an H&E stain or hematoxylin and iosin stain. In these stains, this is a dual stain. So you have a hematoxylin stain that stains the cell nuclei blue or purple. So those are these kind of brown structures in both of these images on the bottom. And then iosin non-specifically stains proteins pink. And so I say non-specifically because this stain is not really going to provide much kind of chemical or molecularly specific information. It's really mainly used to look at cell shape. You know, are the cells round? Are they kind of not round? Are they, you know, perturbed or disrupted in any way? This is mainly used to study morphology. There can be other exceptions to that. But in general, pathologists get kind of a morphological diagnosis. And there are certainly some limitations to this. So here's a case where these investigators were looking at spitzoid, different skin cancer lesions. And these can be classified as benign spitz nevi. So spitz nevi are just, you know, normal skin lesions. They aren't cancerous. They aren't going to spread or cause metastasis or anything like that. And then they can also be classified as spitzoid melanoma, so actually truly cancerous and dangerous and need to be resected. Importantly, in looking at a cohort of these samples, unanimous assignment by expert pathologists is really only agreed upon in about 60% of cases. So that's meaning 40% are not agreed upon by experts. Additionally, of the 60% of cases, that classification includes benign, so a spitz nevi, malignant, and a spitzoid melanoma, and then a large classification of indeterminate or intermediate classifications where they're unable to make a definitive call. And this is really important because these lesions are especially abundant or seen a lot in children and present on the face. And so if we need to do a surgery to resect these and it's on the face of a child, obviously it's important to get that call right. You don't want to subject a young child to unnecessary surgeries. So histology obviously can't tell us the whole story, right? We're only looking at morphology. There's 40% of unagreed upon cases. There's a large number of indeterminate cases. And so we know the metabolome offers a much richer molecular profile. We can kind of look at the molecular phenotype, if you will. And this oftentimes will reveal diseases before they're even detectable by conventional kind of histology, looking at cell shapes in the tissue. So the question is, can we use mass spectrometry to then image tissues to get not only image information, but also molecularly specific information? And obviously the answer is yes, otherwise I wouldn't be speaking here today. And so here's kind of a smattering of a few different types of images we can acquire with imaging mass spectrometry. On the left here, you can see a one hemisphere section of a transverse rat brain section. Each one of these different colors in the rat brain tissue is an individual lipid or an individual metabolite. And I think you can appreciate that we're getting a fairly high level of structural detail. So when we show these types of images to our collaborators, they think that this is actually, you know, fluorescence imaging or something like that. We certainly don't have the sensitivity or spatial resolution of a fluorescence imaging technology, but I think we've come a long way in the past 2 decades or so. And so if you look closely, you can maybe even make out a kind of yellow line in the cerebellum at the top of this image, and that's actually the single cell Purkinje layer in the brain. So the spatial resolution is quite good. The fidelity that we're able to get is quite good. The relative brightness of the colors corresponds to the relative abundance of those lipids and metabolites in the tissue. So we can get some relative quantification or relative quantitative information as well. We're also capable of making very high spatial resolution measurements. I'll touch on this later in the talk. We're also capable of making these measurements with much higher throughput than we could maybe a decade or ago. So we're capable of measuring relatively large tissue areas or even image areas that cover 1 million pixels. And I'll touch on kind of some of these advances later in the talk. Okay, so let's kind of take a step back and start with some of the basics of imaging mass spectrometry. So again, I'm going to focus on MALDI. and MALDI stands for matrix-assisted laser desorption ionization. Obviously, there's other types of ionization techniques I mentioned that we could use, but I'll talk about MALDI. How does it work? What's the general imaging workflow that we're using, and what are some important parameters, mass spectrometry parameters to consider? So MALDI, or matrix-assisted laser desorption ionization, was first invented in the mid-1980s. This was invented by Michael Karras and Franz Hillenkamp, and then independently by Koichi Tanaka. They both kind of invented different lines of the MALDI technique. And in roughly 1985, Karras and Hillenkamp, what they did was they took tryptophan, the amino acid tryptophan, and they mixed it into a sample of alanine and analyzed that using a 266 nanometer laser. And the way this worked is the tryptophan actually absorbed the laser energy and helped to transfer that energy to alanine. So alanine did not absorb 266 wavelength laser energy, but the tryptophan kind of acted like a matrix. And so that's where this term matrix comes from. So the tryptophan acted like a matrix, absorbed the energy and gave it to alanine. And they used this method to analyze small peptides. At first, they were looking at things like melatonin. And then a couple of years later, Tanaka and coworkers used an ultrafine metallic powder with a liquid matrix. So this was actually a little bit different. This was an inorganic particle solution in glycerol. And they used this to analyze small proteins. So they kind of took this to the next level. And rather than analyzing something around 3 kilodaltons, they were using a 34 kilodalton protein. And so Karras and Hillenkamp would later use nicotinic acid and a few other types of small organic acids that have become common today to analyze larger proteins like albumin. And most MALDI matrices today are organic acids, cinnamic acid derivatives, which I'll show some examples of. But these types of inorganic suspensions and metallic powders are still in use in some cases as well. So Koichi Tanaka would go on to be awarded the Nobel Prize, about a decade later, so in 2002. He shared this with John Fenn for their work in basically allowing the field of mass spectrometry to analyze biological macromolecules. So John Fenn won it for electrospray, and Koichi Tanaka won it for his work with MALDI. And I like to highlight this passage of an article that Koichi Tanaka wrote on his work that eventually allowed him to win the Nobel Prize. And he says that I committed a monumental blunder, which was followed by a series of fortuitous and arbitrary decisions. He used ordinary acetone as an ultrafine metallic powder suspension. And one day, by mistake, he used glycerin instead of the acetone, reasoning it would be wasteful to discard the expensive powder. He decided to use the ruined sample preparation technique anyway. And because the glycerin gradually evaporates in vacuum, he knew it would eventually disappear. So instead of waiting, he decided to speed up the vaporization of the glycerin by continually to irradiate the laser. And then on top of that, because he was impatient and wanted these results as soon as possible, he monitored the acquisition of the TOF spectrum while the laser was firing. So in this way, only after at least 4 factors fell into place could he observe this phenomenon that he had never seen before, that no one had ever seen before, and that he won the Nobel Prize for. So I thought, one, it's kind of interesting to hear experts in the field talk about how sometimes they stumble into discoveries. But I think it's also really important, and I tried to express this to my students, that positioning yourself for serendipity in this fashion, when you see and kind of unexpected result or a new observation, can you investigate it and figure out what's going on, because it could be really important or really exciting. I think that type of creativity and positioning yourself for that type of stuff is really powerful. Okay, so how does MALDI work? So nowadays, we typically take a small organic acid, and as I mentioned, this is really efficient at absorbing the laser light wavelength. So typically we're acting in the UV wavelength region, 355 or 266 nanometers. And this so the matrix allows the absorption of this laser energy, and this causes efficient desorption and ionization from the sample surface. So this generates a plume. The laser kind of irradiates the sample and generates this plume, and this plume contains neutral, positive, and negatively charged matrix species. This also includes electrons and negatively and neutrally charged analyte. but the matrix is the dominant component of this mixture. And that's because the matrix is present in vast excess of whatever analyte you're analyzing. So it's typically maybe 1,000-fold, 10,000-fold in excess of the analyte. So it's the major species doing the absorption. And that's important because then your analyte isn't directly absorbing the laser energy, which could be damaging to that analyte. It could cause it to fragment. So the matrix is acting like an energy buffer here. And this initial mechanism kind of remains a little bit debated. I'll highlight two of the major proposed mechanisms or agreed upon mechanisms in the field, but there's generally accepted to be a secondary step where after this initial ablation event, there's a series of gas phase interactions between the matrix and analyte that transfer the charge back and forth and approach a local thermal equilibrium, and then eventually you detect the gas phase ions from this process. So as I mentioned, the role of the matrix is to absorb the laser energy, convert most of that to heat, which causes the ablation event and the sample to disintegrate. This is what we would call an energy sudden process. So this occurs in roughly 3 nanoseconds. At the pulse width, is about 3 nanoseconds. So this occurs in a relatively short period of time, but this is still slow compared to the rate of intramolecular vibration. So this isn't going to be kind of direct vibrational excitation. And then we have this expansion of the material to dilute gas, which occurs over the period of a few microseconds or so. So again, this is all still a relatively quick process, this MALDI ionization event on the tissue surface. So the two main mechanisms, I'll just breeze through these relatively briefly, are first the cluster model or the lucky survivor model. And so this was originally developed by Karras, and this postulates that ions are largely preformed in the solid state, so in the tissue or on the sample surface of whatever you're analyzing. And the ions that we eventually see at the end of the ionization process are those that have luckily survived all of these gas phase interactions and avoided neutralization. And this is generally accepted to describe how very large clusters are formed in MALDI. But perhaps more applicable to things like metabolomics imaging, lipid imaging, and things like that is what was once known as the photoexcitation pooling model, and which is now termed the CPCD model. And this occurs when you have two neighboring matrix molecules, so shown down here on the left, that have been both raised to the first excited singlet state. So they've both absorbed a single photon from the laser. And they're close enough in proximity that their electronic states can overlap, and then you can pool the energy. So essentially you get transfer of one photon's worth of energy to one matrix molecule, and that goes to a second excited state. And then the other molecule drops back down to the ground state. And really important to this mobility and migration is the proximity and the kind of overlap of the electronic states of the matrix molecules. So this hopping event is very, very quick. So this is roughly 50 picoseconds for these energy hopping events. And this is important because remember we said the excited state lifetime and irradiation time of that primary MALDI event is going to be 1 to 3 nanoseconds or so. So what we need is actually at least 2 pooling events or 3 photons to pool together to give us ionization. And the reason for that is because of the ionization potential of a lot of the MALDI matrices that we use. So the photon energy for many of the different lasers that are used in MALDI, you can kind of see here. So a nitrogen laser or an adinium YAG laser that's been frequency tripled are very common. These are probably the 2 most common MALDI lasers that are used in the field. And you can see they have single photon energies of roughly 3.5 EV or so. You can see in this second table down at the bottom that a lot of the MALDI matrices are, around 8 to 9 EVs in terms of their ionization potential. So a single photon is not going to cause them to become charged and ionized and allow us to detect them in the mass spectrometer. So we need multiple of these pooling events to pool together enough energy to ionize this, this matrix. And this is an example of just a mass spectrum acquired with a MALDI ionization process. I know this is a protein. Obviously, we're focused on metabolomics and lipidomics here, but if anyone has ever seen an electrospray mass spectrum of a protein, you can appreciate that this is very different. So in MALDI mass spectrometry, we tend to only see singly charged ions. We don't observe doubly or triply charged species very often. It is possible, but it's just much less common. And so the mass spectrum tends to look a little bit different. For metabolomics and lipidomics, it tends not to look a ton different because we're generally generating only singly charged species there as well. So I've just listed a bunch of common MALDI matrices. These are typically, as I mentioned, small organic acids. And there you can see that they have a lot of aromatic rings. And so those aromatic rings are important for absorbing the MALDI laser energy. And then they also, again, are typically acids or sometimes bases. And so that allows them to serve as good proton donors or acceptors to then transfer that ionization or that charge to our analytes of interest during the MALDI ionization mechanistic process. Oops. So there's a number of considerations that go into choosing a matrix. I won't talk through a ton of these today, but the MALDI matrix can have a different propensity or ability to induce fragmentation, basically how energetic that charge transfer process can be. They can be solids or liquids, so we could use a liquid matrix, kind of like Koichi Tanaka first used. They could be acids or bases, so depending on whether we're analyzing in positive or negative ion mode, that may affect our matrix selection, and they have different abilities to absorb different wavelengths. Okay, so this is our imaging mass spectrometry workflow. So that's kind of how the MALDI process works, and now I'll talk about how we use MALDI in an imaging fashion. So again, we want to make a spatially localized measurement. So we typically take an organ and we section that, or a tissue or some other type of sample, and we section that thinly to about 10 microns or so and mount those onto a flat substrate like a glass slide. And I'll show an example video of this later. And then we apply our MALDI matrix to the tissue surface. And this is done via a number of different technologies, and I'll talk about some of those as well. And we do that relatively homogeneously across the tissue. We then interrogate the sample with our MALDI laser. So we basically fire at many different points across the tissue surface. At each point, we ablate material, sample that into the instrument, and generate a mass spectrum. So we can do this all over the course of the tissue at every single point or pixel where we're ablating, we're acquiring a mass spectrum. And if we can reconstruct heat maps of intensity for any of the ions that we've detected in the spectrum, we can plot their intensity as a function of position across the tissue surface. And so we end up with these heat maps of metabolic intensity. And so that's kind of what I would call the most conventional imaging mass spectrometry workflow, where we're imaging an entire tissue section or a large area of tissue section. We can also do what's called histology-directed acquisition or profiling imaging mass spectrometry. And so in this case, we would have the tissue first annotated by a tissue pathologist prior to mass spectrometry analysis. And then we would then only inquire information, mass spectrometric information, from small predefined regions of the tissue. So rather than imaging the entire tissue, we're just going to acquire data from select region that the pathologist tells us of interest. This may be cancerous, this is indeterminate. So we're just acquiring spectra from these regions. We can then do statistical classifications to compare, the metabolic profile in this region to this region to say, okay, these are truly different cells, or maybe they're the same cell. And so that can allow us to classify regions of the tissue. Okay, so when we're setting up an imaging experiment, there's a number of figures of merit that we need to be considered with. Some of these are very traditional mass spectrometry, kind of classical figures of merit, things like mass resolving power and mass accuracy, sensitivity. But there's also a few considerations that are important, that are kind of unique to the imaging experiment. And one of these is spatial resolution. So the distance between two adjacent pixels. And this eventually determines the level of structural detail that we can glean from an image. And then the throughput and file size is really important as well. So as I mentioned, we're taking many different measurements across the tissue surface. This is typically thousands of individual measurements. So the time per pixel, the time per mass spectrometric acquisition needs to be very rapid. Otherwise, we're going to spend days analyzing the sample. And then obviously, if we're analyzing thousands of pixels, the file size can become important as well. So it's not uncommon for some of the high performing mass spectrometers to acquire file sizes, hundreds of gigabytes in size, maybe even a terabyte. So we have to choose our experimental parameters wisely. And I say we need to choose them wisely because we do have some flexibility in how we set these things up. And we often discuss this, it makes the most sense to discuss this in terms of spatial resolution. So when I go to our collaborators and they're interested in doing an imaging experiment, they typically say, I want the best spatial resolution possible. I want, 10 micron spatial resolution or 1 micron spatial resolution because they want the prettiest pictures. But oftentimes that's not necessary to answer the biological or clinical question at hand. And that's important because as we decrease the size of the incident laser beam, the amount of material we ablate goes down. So our sensitivity decreases. And additionally, the time required to sample a given area and the file size increase dramatically. So we typically kind of tune this slider bar to meet the tasks of our experiment at hand. So I'll kind of highlight a few different aspects of some of these parameters over the next, remaining 30 minutes or so. Okay, so let's talk about some sample preparation. The entire procedure from acquiring the sample, through sectioning the sample, through any washing of the tissue to matrix application is critical. And this is not unique to imaging. This is obviously very true for classical metabolomics as well. But for imaging, we really want to maintain the molecular and spatial integrity on the tissue surface. So we want to minimize any chemical degradation of the metabolites. We want to minimize any kind of walking around or movement of the metabolites in the tissue section, which can cause basically physical deformation of the image. And then obviously we want to optimize the sensitivity of the experiment. So imaging mass spectrometry, I think, probably works best in this kind of intermediate spatial resolution range. We can certainly image an entire animal or a cross section of an animal. So this is a relatively large amount of tissue area. It's possible, but it would take a long time experimentally. We can also analyze single cells, and this has been done in imaging mass spectrometry. But these are quite small and typically challenge the spatial resolutions of most commercial instruments. So I think the sweet spot for many imaging mass spectrometry experiments is looking at intact organs or tissue biopsies, as you'll see. So one of the most important things to consider when we start the experiment is the type of sample that we get. So we prefer to use fresh frozen tissue. So we freeze this typically as quickly as possible using dry ice or maybe liquid nitrogen. And this tends to be the easiest type of tissue to work with, especially for metabolomics and lipidomics experiments. There are obviously a large number of archived and embedded samples that are available in the clinical field. So these are typically FFPE tissue samples, formalin fixed paraffin embedded tissues. And it's possible to use these tissues for imaging mass spectrometry. But typically the proteins are cross-linked. And so it's difficult to obviously analyze the proteins. You need to kind of free them from that chemical cross-linking to do so. And additionally, in order to deparaffinize the samples to analyze them, you're subjecting the tissue to substantial washes. And so this could lead to some solubility and delocalization of some of the small metabolic compounds in the tissue. So these just tend to be more difficult to work with. So we try to avoid using embedded samples if possible. Some other things to consider are variability over long-term studies. So changes in personnel, equipment, instrumentation parameters. These are all things, that are important to consider when setting up kind of a cohort of imaging mass spectrometry experiments. Okay, so I'll show a video of how we section these tissues in just a second. There's a few different types of cryostats or cryomicrotomes that you can use. Our lab uses a Leica CM3050S, and you can see kind of in a picture of the interior chamber here on the bottom left, and on the bottom right is kind of a close-up zoom in of this central circle portion of the middle figure. So this is what we call the cryostat head. I think these labels have walked around a little bit, but the central region here is the cryostat head that holds a chuck that holds the tissue. And so this tissue is being sectioned on a blade that has been inserted into this stainless steel stage here. And you can see that this paintbrush is peeling off a roughly 10 micron thick tissue section that we can then mount onto a microscope slide. So Chelsea and Julia here are going to demonstrate this process. So you can see that the tissue section is being moved down across the cryostat blade, and that pulls off a very thin tissue section. So this essentially functions as a very precise deli slicer. So there's kind of a wheel on the side that you crank, which will move that head up and down and advance it close to the blade. We then use some artist paintbrushes to remove the tissue section and position it onto a Teflon coated slide, which Chelsea is doing at the moment. So she's making sure there's no folds or tears in the tissue as she positions it. And then she's going to take our microscope slide, ensuring that it's free of particulates. And then she's going to basically pick up that tissue off of the Teflon slide. So she's picked it up, and then we perform a thaw mounting procedure where we essentially warm the back of the slide with our hand, and that ensures that the tissue adheres well to the slide. Okay, so that's how we kind of acquire and section the tissues. We then need to obviously apply a MALDI matrix surface or coating to the sample, and so there's a few different ways that we can do this. In general, this is always a balance between analyte extraction and analyte delocalization. So when I say extraction, we're applying a MALDI matrix to the tissue surface, and we need the analyte to be extracted from the tissue, so whatever metabolite we're interested in, and form co-crystals with the matrix. So that enables, when we fire the MALDI laser, that enables for the analyte to join in the ablation event and be detected in the mass spectrometer. So this extraction process is very important. Sometimes that extraction can occur via very dry matrix application technique, and sometimes it needs to be occur in kind of a solvated environment to help facilitate extraction into the multi-matrix layer. Now, if it's too wet, if that environment is too hydrated or there's too much solvent present, then we can get delocalization. So on the left here, you'll see that we've applied a very large amount of liquid to help with extraction, but because there's so much liquid, the analytes can kind of move around spatially. So we've lost the spatial integrity of the analytes. So when we make our MALDI imaging measurement, it's not going to be representative of the true biological distribution of some of those metabolites. So again, this is a balance between enough solvent to encourage extraction, but not so much as to facilitate delocalization. So there's a few different ways that we can apply the matrix. We can do this with just a basically manual pipette technology using a simple pipetter. And this results in very large matrix spots. And this is useful for some of those profiling experiments that I mentioned earlier. But if we want to do kind of true imaging, we need to apply smaller droplets, such as you see here in the middle, or true homogeneous coatings across the tissue surface. And so for manual spotting, as I mentioned, we can do this with a classical pipetter. This is great. It gives very good extraction because it's a very hydrated environment. So it's very good for sensitivity, but it's quite poor in terms of the spatial resolution. Obviously, we're plopping down very large droplets, so we wouldn't be able to image over the entire tissue section. So kind of an intermediate between a manual spotting approach and a true homogeneous coating is a robotic spotting approach. So this applies much smaller droplets using maybe acoustic spotting or acoustic transducer technology to the tissue surface. And so these spots are 100 to 300 microns, which is still relatively large, but can enable some degree of imaging for the tissue surface. There are other types of technologies as well. So you could use a modified inkjet printer to apply a MALDI matrix. You can use a manual spraying approach using an artist's paintbrush or a TLC sprayer. And these are relatively kind of inexpensive DIY approaches, maybe around less than $200 to make these modifications and apply matrix. But probably the two most common forms of matrix application in the field are sublimation and robotic spraying technologies. So the sublimation setup is shown here on the right. This is something that you can build for about $5,000 in your lab. If you already have a rotary vane pump, That's the main expense associated with this setup. So if you already have that, you could probably build this for less than $2,000. So this is what I recommend folks who are just starting imaging to try and start with. This is a very dry approach. So you're just using a sublimation to generate a gaseous form of a MALDI matrix and deposit that onto a tissue surface. But it's very good for high spatial resolution. It still is quite good in terms of sensitivity, and that very dry nature can be combated with some downstream sample preparation effects. If you're a relatively high throughput lab and you're doing a lot of matrix applications and you want to be very reproducible and very controlled, you can purchase a robotic sprayer. There's a few different manufacturers who makes these. HTX Imaging is one. Bruker makes one. There's a few others. Sunchrome Suncollect makes one. And these are really great. They're very reproducible, great sensitivity, but they're much more expensive. You know, they're typically $60,000 to $80,000. So I'll show an example of this robotic sprayer here. So this is Chelsea and Julia again. And so you'll see that the matrix solution is passed through this nebulizing head that's moving back and forth across the tissue, across the microscope slide with the tissue on it. And you can see this yellow matrix that's being sprayed onto the tissue surface. So obviously, since this is a sprayed solution, it's a little bit wetter. So we tend to get a little bit better abstraction of the analyte into the tissue. But again, we have to be careful not to have it so wet that we actually induce delocalization. Okay, so some of the considerations of the matrix selection. We can optimize the identity and the solution of the matrix to basically facilitate the best abstraction. So some of the matrix form different crystal sizes than others. Some of them are better for certain classes of analytes, positive and negative mode. So there's kind of a whole database of information out there where you can look up some of these things. And a lot of it is also done empirically. So when we start a new experiment or a new project, typically we'll screen a matrices that are appropriate for this. Additionally, recrystallization can be used as well. So I mentioned that even with dry matrix application techniques, we can facilitate extraction after we've done the matrix application step. And so that involves, once we've applied the MALDI matrix coating onto the sample, we can incubate the sample in a relatively hydrated environment for a short period of time to help with that extraction. And then we can also do some tissue section washing prior to matrix application that I won't really talk about here today. Okay, so let's talk a little bit about spatial resolution. So this is the image resolution that we end up with from our resulting experiment. And this is important because it determines the level of structural detail. So at higher spatial resolutions, we can look at smaller and smaller structures. In this picture here, I'm showing a few images of pancreatic islets, and you can see it's not till we get to about maybe 20 or 30 microns can we actually fully resolve these islets. So some biological questions can only be answered at high resolution, but it's certainly not always necessary. So some of the things that affect spatial resolution, I mentioned already that our sample preparation can certainly affect this. So the crystal size or the spot size of what we're applying can determine the spatial resolution. But also the mass spectrometer can limit the spatial resolution as well. And so this could be the laser optics or how finely down we can focus our incident laser beam, the ability to move the stage, which moves to the next pixel position, as well as the signal intensity. If we have a very small laser beam and we're not getting any analytical signal from the tissue surface, obviously we're not going to be able to image that. So there's a few different regimes that we can sample the tissue at. One is sampling at the laser resolution. So let's say we have 100 micron spot size. We can essentially move the laser beam 100 microns using the stage motion and perform imaging in that fashion, which works perfectly well. We can also do an undersampling or profiling approach where we're just analyzing at a larger diameter, and that is perfectly suited as well. And then we can also do what's called an oversampling approach, where we move the laser beam at a fraction of the beam diameter. And essentially then the new, basically the new material from each subsequent ablated pixel is only coming from a fractional kind of hemispherical or half moon shape of the leading edge of that laser. And this is a way to basically get spatial resolutions smaller than the beam diameter. So here's an example of us acquiring an image. Chelsea and Julia, you can see in this upper left-hand picture of the camera here, the laser is firing every roughly second or so, and it is generating a different mass spectrum at each point. So this is a relatively slow acquisition that we're obtaining at the moment. And you can see the spectrum is updating every second or so. We're looking at some different metabolites, fatty acids, and lipids here. You can see the progress. We're about maybe 2/3 or 3/4 through the sample. This was acquired on a Bruker Solarix FTICR mass spectrometer. And again, you can kind of see the live camera view of the laser firing roughly every second. So I'll kind of fast forward here. So this is some more spectra updating. These are some phospholipids, some phosphatidylcholines actually that we were analyzing. And you can obviously zoom in and look at different regions of the spectrum. Okay, so there's a number of different ways that we can render the images or look at them. There's a lot of different vendor-specific softwares and freeware out there that you can use for this. I'm showing an example here using Flex Imaging, which is Bruker's imaging software. So we can definitely select different peaks, different M/Z values in our spectrum and show them as a false color image. So in this rat brain tissue section, I'm just clicking on different M/Z values and looking at their spatial distribution in the tissue. Okay, so high spatial resolution again can be important depending on the task that you're trying to achieve. And I won't spend a ton of time talking about this. There's a lot of effort in the field about looking at different ways that we can improve spatial resolution. So there's a lot of hardware research that's going on at improving that. This was an image that was acquired during my postdoc using one of those kind of advanced hardware setups. This is a human kidney glomerulus. This is a lipid and metabolite imaging experiment. In red here, you can see the kidney glomerulus surrounded in blue and turquoise by proximal and distal tubules. And you can maybe make out these little black flecks in these green and blue structures, and those are actually individual cell nuclei, which is confirmed using a PAS counter stain. And so again, this is a kind of heroic imaging experiment, but an example that some imaging mass spectrometry experiments can approach single cell and even subcellular spatial resolution. So this is just another kind of snapshot of a lipid and metabolite imaging experiment of a human kidney as well, or excuse me, a mouse kidney or a rat kidney as well. And you can see different regions of the kidney are differentiated. Again, each color is a different lipid or a different metabolite, and the relative brightness corresponds to the relative abundance of that. So not only are we getting spatial information, but we're getting some molecular information as well, looking at each of these lipid channels. Again, here's a brain image acquired at relatively high spatial resolution, and we can get quite nice structural detail. Okay, so let's talk a little bit about the importance of molecular specificity. I mentioned that we're getting some chemical information, so differentiating between the many isobaric and isomeric compounds in the sample is really important, especially in metabolomics and lipidomics experiments. What I'm showing here is an average mass spectrum acquired from a rat brain tissue experiment. And this is the kind of lipid mass range and maybe the high end of the metabolomic mass range. And you might say at first glance, okay, there's maybe a few dozen peaks in this mass spectrum. But if we were to blow up the y-axis, there's actually several thousand peaks. And if we were to zoom in on a very small half Dalton range of the x-axis, you can see that even in this small range, there's maybe a dozen peaks or so with quite good signal to noise. And again, if we take each peak and plot the spatial distribution of it, we get a mosaic of several hundred or thousand individual ion species. And each of these could have an interesting spatial distribution or a different spatial distribution. So really differentiating between all of these peaks is really important. And one of the ways we can do that is through the use of high resolution, high mass resolution. So here's an example of a pancreas sample, and I'm just showing the distribution of each of these three peaks in this roughly 70 millidalton mass range. And you can see that each one of these has a different chemical identity and a unique spatial distribution. So we need sufficient ways to differentiate between these peaks. If we didn't have enough mass resolution, this would look as a kind of one single blob, and we would look at a composite image. And so that's kind of what I'm showing here. If each of these different color traces are different mass resolutions of a single peak. And it's only when we get to maybe 420,000 resolving power or so that we have enough resolution to resolve all of the peaks underneath this peak. And so again, if we didn't have that, we wouldn't be looking at the true informative chemical image. So that's great, but this high mass resolution is expensive. These types of instruments, FTICR and Orbitrap instruments, are relatively expensive, and it comes at a significant cost per time. And additionally, this resolution is going to separate true isomers. So we need other ways of separating these chemical compounds from one another. And one of the ways that we can do that, again, is through accurate mass measurements and high resolution. We can also use ion mobility spectrometry, which you may have heard about, and then we can also use tandem mass spectrometry. So here's kind of another example looking at a few different lipids that, again, show different spatial resolutions that are still relatively close in mass. So we would call these isobaric compounds because they have the same nominal mass of 888, but a different fractional mass. Here's an example where we're using tandem mass spectrometry, which you've heard about today. So fragmenting the lipids to look at different portions of the fragments. And this can allow us to differentiate exact isomers from one another. So here we're looking at 2 phosphatidylethanolamines, 36,2, so 36 carbons in the fatty acyl tails, and 2 double bonds. This ion population has 2 different isomers, an 18,0, 18,2 isomer and an 18,1, 18,1. And if we do the tandem mass spectrometry imaging experiment, we can see that they have different spatial localization. So again, it's important to be able to differentiate these. This is especially important in imaging mass spectrometry because we don't have the benefit of an upfront separation like GC or LC. We're limited to only the ion types we generate from the surface, and we don't have the ability to do some kind of advanced separation like we would otherwise. And given that there's nearly 100,000 possible lipid structures, and there's a lot of structural homology in the metabolome, this is really important. So I have a couple other examples here, but in the interest of time, I think I'll kind of pass over these. There's some other types of MS imaging experiments that we've done that show that this is important. My lab in particular is looking at new types of gas phase approaches that we can use to allow us to differentiate these things. I'm going to skip past this though. I'll show a spectrum at the end that I think is quite interesting. So using this gas phase approach, we've actually been able to show that a single phosphatidylcholine lipid is actually made up of at least 5 different isomer populations, depending on the fatty acyl components. And I say at least 5 because there's also double bond isomers to consider, like we first talked about in the fatty acid breast cancer project at the beginning. So again, it's important that we continue to develop technologies to differentiate these. Okay, so I'm running a little low here on time, so I'll talk about a few applications in brief. I'll just say that we can perform drug imaging and that there's ways that we can do quantitative imaging. This is important in the pharmaceutical industry, obviously, because we want to monitor different absorption, distribution, metabolism, excretion profiles of drug compounds as they go throughout the development pipeline. Typically, this is done with something like LCMS, but again, we lose a spatial context with the LCMS measurement. And so we'd like to be able to spatially monitor where the drug goes throughout its development in the body or the body of an animal, as well as how much of it is there. And so we can do quantitative imaging, mass spectrometry. I won't talk about a ton how we do this, but we can generate calibration curves on control tissues and then use that to figure out how much is actually in a dose tissue. We've also used imaging mass spectrometry to look at infectious disease. So this is obviously an increasing problem in the scientific community and in our society as well, antibiotic resistance, and during bacterial infection. There's often an interplay between the host immune response as well as the invading pathogen. And so we've been interested, and our collaborators have been interested at looking at the metabolic crosstalk or interplay between those two regions. So looking at how the infectious agent responds to the host immune cell. And so this is an example where we're looking at systemic staph infection that has created abscesses in these soft tissues. And we've looked at kind of the anatomy of the infectious abscess to see if we can get some information about what is going on. Some of the things we've looked at are calprotectin proteins, which are nutrient metal binding proteins, which sequester some of the nutrient metals that are required for metabolic turnover in the bacteria. We've done this on a number of different animals, number of different organs. Kidney is very popular in terms of looking at staph abscesses. And we've noticed that there's some heterogeneity within individual abscesses. So you can see just in the color schemes here that there's different molecules and different intensities present. So what we've done is performed a three-dimensional imaging analysis of this. And so rather than looking at just a single 2D slice, we've looked at many serial two-dimensional slices through the organ to kind of section through and look at some of those infectious foci. And for this, throughput is a very real concern, obviously, if we're looking at all of these tissue sections. So I'm going to fast forward here and show a couple videos of this. So here's an example of a relatively high throughput laser. So on the left, you'll see kind of that conventional acquisition speed I was showing earlier. And some of the new technology is shown on the right, where we're imaging and the laser is firing at roughly 20 kilohertz or so instead of a single hertz. So the new technology can sample really rapidly. We were able to use that to analyze many different planes through a kidney section. So you can see the kidney down here in yellow, the abscess is shown in blue, and you can see that we're sectioning through the abscess. And in different regions of these abscess, you'll see this kind of donut hole formation. And in the middle of those donut holes are active bacteria that are proliferating. And as you can see that these different abscesses look different at different stages through the tissue section. So we're kind of trying to study the holistic anatomy of this infection. Okay, so in the last minute or two here, I'll breeze through a couple other applications. We're also interested in studying diabetes. So another important health problem in our field, or excuse me, in many different fields, both in type 1 and type 2 diabetes, a lot of research has been done, but a lot is still unknown. So in type 1 diabetes, you suffer basically an autoimmune process is triggered that causes a progressive loss of beta cells. It's not really known what this is. And even in individuals who have had type 1 diabetes for decades, there are some beta cells that persist, but it's really unknown what is causing these things to persist or what's allowing these beta cells or insulin positive cells to survive. Additionally, in type 2 diabetes, there's a lot of molecular heterogeneity in this disease and even phenotypic heterogeneity. Some beta cells suffer from amyloid plaques and some do not. And the pancreas in particular is a challenge to study because the islets or the important hormone producing centers are roughly only 1% to 2% of the mass of the entire pancreas. So it's really important to have some spatial measurements with this. Additionally, a lot of the mouse models we use to study diabetes are problematic because they don't accurately recapitulate the human anatomy. So we've used imaging mass spectrometry to study different samples and looked at different lipids and metabolites. Again, subtle chemical differences result in dramatic differences in spatial resolution. I don't have time to kind of go into the details here, but we've looked at many different kind of signaling gangliosides, and they appear differentially expressed in different regions of the islets, as well as differentially expressed between the mouse and the human samples, which obviously gives some evidence that there's molecular evidence or molecular differences between the mouse and the human. I'll also say that there's a lot of computational aspects that are going into imaging at the moment. One area is image fusion, where different imaging modalities are being mixed and fused together. So imaging mass spectrometry and many classical immunochemistry microscopy technologies are kind of being used and mathematically fused together to give informative images that combine the molecular specificity of mass spectrometry with the spatial resolution of classical microscopy to give you the benefits of both technologies. And kind of finally, I'll kind of wrap up by concluding where we started. I said that, in a lot of these clinical studies, we have non-unanimous agreement and indeterminate cases. Well, we've used imaging mass spectrometry to study some of these cases. In many cases, the IMS diagnosis backs up the histopathological diagnosis, and this is supported by the clinical status, the clinical follow-up for many years. And then there are cases where the imaging mass spectrometry analysis disagrees with the histopathological diagnosis. So here you can see that the pathologist said that this tissue or this lesion was a melanoma. The IMS said that this was a benign Spitz nevi, and the clinical status actually backs up the mass spectrometry diagnosis. So it shows better accuracy. So the clinical status that each of these individuals was alive with no evidence of disease, even up to several decades past the initial diagnosis. And then for those indeterminate cases, the IMS was able to make a definitive call. And again, the clinical follow-up backed up the mass spec diagnosis. So not that mass spectrometry is going to replace a pathologist, but it's another tool in their toolbox to hopefully improve their specificity and accuracy. So when we're doing data analysis, we can use statistical tools to classify the metabolic makeup or profile of some of these tissue samples to make classifications. But we can also look at kind of a more holistic biochemical profile or pathway of the metabolites that we're detecting to see which pathways are elevated in certain disease states, to then hopefully get some information about the disease progression. Okay, so I know I kind of zipped through the past 30 slides or so, and I'm a little bit over time, but hopefully I've convinced you that imaging mass spectrometry can enable us to visualize biochemical processes directly in tissue specimens with high fidelity. And it provides a really high degree of molecular specificity that you don't get with a classical imaging technology. So it's the best of metabolomics that we're learning about, but we're also doing it in an imaging fashion. And these approaches are really useful in a wide variety of areas. So with that, I'll stop. If we have time for any questions, I'd be happy to take them or answer them in the chat if we need to move on.
Sure, thank you so much, Boone. And there are some very interesting questions. So there are two from Dave. One is it more correct to think of plumbed matrix as a gas sheath, as in some welding applications, or as a normalization structure like in cryo-EM?
Yeah, so let me see, the plume gas as a gas sheath. So it's kind of thought of as like a plasma. Yeah, so I think that's kind of what you're getting at. Like it's in the gas phase, but it's a relatively dense gas phase. So it's kind of considered like a little local plasma, and it approaches a local thermal equilibrium because the density is so high. But that occurs really quickly. So it's not something that lasts for longer than a few microseconds at best.
Next one, is matrix photon pooling analogous to EM gain chip of EM CCD cameras?
I'm not familiar with that exact reference, but if it's familiar with CCD cameras that I've kind of been familiar with in some of our field stuff, it's not completely dissimilar. I mean, it's a little bit different in that some of these cameras are basically getting impinged in the same spot, in the same basically the same pixel, whereas you can think of this energy pooling as kind of combining different regions or different pixels or different aspects of the camera to give a single intensity out of one of the matrix molecules. So it's kind of like a spatial aggregation of the photons into a single point.
Yeah, the next one is about whether imaging can be applied to plant tissues. The answer, of course, is yes. But yes. Could you comment on how to overcome the cell wall barrier and what matrix you would suggest to use for plant tissues?
Yeah, so Dr. Chen knows this very well. We collaborated on some of this stuff. So yes, the answer is yes. It's been used in a wide variety of areas. It's actually really popular in some of the Midwest universities. They're looking at things like corn crops and things like that in a lot of the agricultural universities. But we do some of that here too, obviously. And the cell wall can be challenging depending on what you need to analyze. Obviously, that waxy film on a lot of leaves and things like that can affect your analyte extraction. So sometimes we use some solvents or things like that to disrupt or dissolve or digest some of the components depending on what we need to analyze. And the matrix that is best suitable just depends on what we're looking at. So Dr. Chen and I are looking at glycans, so we've tended to use something like DHB or CHCA. If you're looking at lipids, you tend to use something like DAN or 9AA. So it really just depends on your analyte class.
Great. There's another one. How does laser influence tissue in hydrated and dry conditions?
Yeah, so there's two aspects of that question. Generally the tissues that we look at are dehydrated and dry. So that thaw mounting process tends to remove most of the solvent. So it tends to be a fairly dry environment by the time we get it into the mass spectrometer. And then when it's in the mass spectrometer, it's obviously in a vacuum environment. So it's going to be quite dry. But the other side of that is, does the laser basically affect the tissue sample? And so since we have a matrix coating on the tissue, the matrix is absorbing a lot of that laser energy. So it doesn't actually do anything destructive to the tissue surface. So we can take that tissue, apply matrix and analyze it again. We can take the tissue, wash off the matrix and do histological stains. So the tissue it's not really a destructive analytical technology.
And another question is, how internal reference is defined for quantification of imaging mass spectrometry?
Yeah, that's definitely important for quantification. And so there's different ways that we can apply it. We can apply it if we're doing, for example, a robotic spraying approach. We can spray the internal standard across the tissue. If we're doing a very precise spotting approach, we can spot internal standard on the tissue. So there's different ways that we can do it. It just depends on the spatial resolution that's required. You really want the internal standard to kind of match the analyte in the tissue sample in terms of how it's being extracted into the matrix layer. So for example, we wouldn't mix the analyte and matrix and then spray them, because then they're already co-crystallized, whereas the analyte is not. So we'll typically apply the internal standard and then maybe apply the matrix, so they have equal opportunities for extraction.
Another question is, could one combine storm paint with the imaging mass spectrometry?
So storm paint, I'm not familiar with.
Yeah, neither do I.
If that person wants to clarify in the chat, I'd be happy to answer that. But I'm not. Storm, I believe, is like a. I believe it's a microscopic imaging technology, but I'm not familiar with it enough to answer that question.
Yeah, maybe you can find out and put in chat. Let's have the last question for this session. Is about considering double bond positions. Is it a viable thought to introduce techniques similar to UV photo dissociation in MALDI 2 fashion for improvement in lipid structure resolution? How challenging technically such a setup would it be?
The answer is absolutely. So my lab and another number of other labs are doing things exactly like that right now. We're doing UVPD. We actually are doing electron-induced dissociation. We're doing some of these ion-ion reactions. I briefly mentioned ion molecule reactions. So that we're exploring a lot of those avenues for metabolite and lipid ID. Technically, 10 or 20 years ago, they were super challenging. They're still a little bit challenging today, but a lot of the instruments come capable with UVPD now. It's actually commercially available. Or ion-ion reactions such as electron transfer dissociation. So the hardware modifications required for this are a little bit easier to manage nowadays than they were 10 or 20 years ago. So technically, I guess it depends on your abilities, but it definitely can and is being done.
Great. Let's thank Dr. Boone Prentice again for such a really nice talk and great discussions. So Boone, if you could address that storm question, if you find out in the chat, that'd be great.
Will do.
Thank you very much. So let's move on. Our next speaker is Dr.